My name is Meredith Hartley, and I'm an assistant professor in the KU chemistry department. I'm really kind of positioned both in bioanalytical chemistry and also more in biological chemistry. So we study the process of myelination, and so myelin is this fatty substance in our brains that surrounds axons, and it acts basically like rubber on an electrical wire to enhance the speed of signals being sent by the neurons in our brain. And so we're really interested in that substance because it's really important for brain function, and because when it's damaged, that results in various diseases like multiple sclerosis, for example. But there's also a lot of myelin damage in Alzheimer's disease and other neuromotor disorders. And so what we're interested in about myelin is that it's a very lipid rich substance. And so when myelin is damaged, that means that all of these lipids are released into the brain or into the spinal cord. And then the tissue has to find a way to deal with that, deal with all of those excess lipids, recycle them and put them back into new myelin that can be formed. And so we are using a variety of methods and approaches to kind of answering those questions and trying to identify pathways of lipid recycling and myelination. But I have a lab of graduate students who actually do this research. Each of them has a project focused on kind of one aspect of what we're interested in, or sometimes multiple projects, and they really run the show. So they're the ones doing the experiments, analyzing the data, and I get to just kind of watch from the sidelines and help, I hope, some of the time. It seems like most of the students in my lab are interested in industry positions, ultimately. But you know, I do have one or two students who are interested in more in teaching aspects of an education. We use a couple of different core facilities that are important for our research. The first one that we use probably the most frequently is an imaging core facility that's located on the medical campus in Kansas City, the KU MC. And that is where we do a lot of fluorescence microscopy using a spinning disc confocal microscope there. The other main core facility that we use is the flow cytometry core, which is housed in the molecular biosciences department. And we use that to take our brain tissue samples and separate it into kind of single cell populations and then characterize those cells.